going to wrap up today, reflect on some of the things I've heard, and then, open it for some discussion. But first I thought I'd just say, thanks very much to everybody that has contributed to the presentations, and obviously the wider team of people that contributed to all the presentations, and David for leading this, being the driving force behind getting the funding for the proposal, and then leading it expertly over 5 years, keeping everyone together and on track, and mentoring 3 PhD students and lots of other early mid-career researchers through the process. I think it's been very beneficial in that sense, and obviously we've seen today some pretty really interesting science as well. It was very nice to see it all come together. So what I've done is just written a few points that I took from each of the talks today. Very few. It's quite hard to do actually. And so I'll go through those, and then we'll just open up for discussion. And particularly I'd be really interested in the future opportunities. So it's kind of like we've found some cool stuff through this research, but where are we going? Where are the unanswered questions that we can try and tackle in the next 5 years? So right at the start, David, I guess, framed the challenges, particularly around the dairy expansion on the stony soils in Canterbury, and that recoupling carbon and nitrogen could help mitigate the unwanted losses from some of these systems. One thing that I thought was really interesting, and David referred to this, was that on one of those really early slides he had headroom. And I think in the last 5 years, that is a word that's gone completely out of vocabulary. I haven't heard it for a very long time. So quite an interesting reflection on how things have changed. And another thing that David emphasises is that the program was structured across scales. So we were going from microbes, which we heard about from Gwen and Andrew, right through to a lot of work on lysimeters, at different scales of lysimeters, through to the paddock. And then I thought it was really interesting that Hirani, who was next, he said a couple of times, ecosystem boundaries are not farm boundaries. So there he was taking that scale out even further, which was quite interesting. So Hirani, he's gone, has he? Yeah. He gave a nice overview of the Moriora systems framework, and it's kind of a decision-making framework grounded in Te Ao Māori. And it seemed that protecting Mahinga Kai was a key motivating factor behind the development of that framework. William then gave us a very dense summary of on forage crop and pasture swards impacts on nitrogen leaching, and I was struck by how much he must have done in his PhD, because there's a lot of treatments and I couldn't keep up with them all. He looked at different forage crops, sward types, urine loading rates, carbon inputs, and in winter growth. They all have a really big impact on N leaching. And then I think one of the challenges or opportunities that he identified was fodder beet. What's going on with a potential biological nitrification inhibitor is something to look into in future. Scott looked at the carbon and nitrogen inputs and losses under lucerne, and obviously irrigation, 70% increase in forage production, but it didn't lead to an increase in carbon in the system, which was quite interesting. And then largely because that's cut and carry, and so a lot of the carbon was taken out of that system. And when it was grazed, you actually retained more carbon in the system, but a lot more nitrogen losses. So trade-offs. Andrew and Gwen covered microbial regulation of carbon and nitrogen cycling. One of the things that they emphasised was that carbon inputs had a pretty large and long-lasting impact on fungi in the systems. They also pointed out that there was lots of microbial diversity at depth, and that's missed if we only sample in the topsoil, and that the deep soil communities are more connected than the topsoils, possibly because of resource limitation or clusters of resources and the microbes are more connected. Andrea gave us an overview of plant species effect on carbon rhizodeposition, and she showed that first figure where there was a really clear separation in the microbial community composition between ryegrass and plantain. And then one of the other bits that she showed was that under the ryegrass, more carbon was taken up by the fast carbon cycling groups, and the opposite, and the carbon was taken up by the slow carbon cycling groups under plantain. Then we changed tack again quite dramatically to phosphorus leaching under lucerne, and Rich reminded us that we can't forget about phosphorus. It is a nutrient and it's actually quite important in relation to water quality. We tend to be quite focused on nitrogen. His results showed that there was no differences in P concentrations in the drainage water of any of the fractions, but there was more loss in total because there was more drainage. And then he showed the quite startling figure from the Lincoln University dairy farm, and he suggests that the phosphorus losses increased dramatically over time, and so he encouraged us, if we can, to keep monitoring, because as Olsen P increases at Ashley Dean, as it beds in as a dairy farm, losses could get much greater. And then Joe looked at modelling the effects of lucerne management on carbon and nitrogen trade-offs. As David said, this was kind of a culmination of the project, really, or lots of pieces of the project, taking a good chunk of the data that we generated within the project, using that to validate and test the model, and then running a whole bunch of different scenarios so that we could see where that optimum was. And that's hopefully will be a practical thing that farmers can use to provide advice for farmers around management options. And then Johannes talked about paddock scale nitrous oxide from grasslands. Provided some pretty good evidence around the split footprint approach that was quite encouraging, that seems to work. So we can actually look with one system, look at two different treatments. It was quite obvious that the ryegrass clover pasture actually gained quite a lot of carbon in that system, unlike the lucerne previously. But the lower under the ryegrass, the mixed sward, it wasn't, and that was possibly because of one heavy grazing. So it'll be interesting to see what happens over this spring. And there was lower N2O from the mixed swards during periods when the cows were grazing. So again, it'll be really interesting to see what happens over this season. But then he said, overall, if we consider both CO2 and N2O and the greenhouse gas effect of those, the ryegrass clover is better. And then finally, Andrew gave us an overview on the Carbon Watch program, and that was really taking scale to the next level. Instead of going from microbes through to lysimeters through to paddocks and then maybe the local waterway, it was taking it to the national scale. So it was quite cool to see how all of the lots of different pieces of data came together. It was a pretty ambitious project, and I think that question at the end was a good one. I'll be very interested to see how it all comes together in a couple of years. So that was kind of my very quick summary of some of the points that I took out of the day. Does anyone else want to just reflect on any key points and things that they'd kind of like to restate or emphasise, or that they really enjoyed in the first instance? Anything that someone wants to add? So, say on what the net greenhouse gas effect is of going from dairy farms to such a cropping system. Yes, we will probably lose a good chunk of soil carbon, but you're also losing methane. And so we need to get, I guess, really good numbers to go, this is what's going to happen, so that we can make informed decisions about those. And then I guess there is potentially scope within those cropping systems to, or alternative systems, to make them so that they don't, you don't lose soil organic matter potentially.
Thank you. I think there's differences in public perception in terms of social license, isn't there? That greenhouse gases is largely, everyone knows there's an issue, but it's not going to be bad for us tomorrow, and people can't do about it. But when we look at water quality, that is really directly affecting people. It's their drinking water and the health of their people and their children. So there's much bigger emphasis on it. And every day in the newspaper there is something about water quality in Canterbury. So we know it's there. And I guess this programme and what we're doing, at least it's provided numbers we didn't have before. So we know about inputs and losses, leaching possibilities, and that's been of value. But the other point I want to make is that, Keith Wood was talking on Monday on Catherine Ryan's programme about the value of carbon, and it seems that the value of carbon in wood is way above the limit. It's now up to $70 a tonne CO2, which has never been expected before. But soil carbon doesn't have any value at the moment, and that's largely because we can't measure it. It's very difficult to measure. And as we've shown, we can using our techniques, we can have a good indication of what's happening in relation to a particular treatment, but we can't necessarily do that everywhere across country, let alone across New Zealand. But if we can get to the time where people value soil carbon, and it's seen to be something to strive for because of the benefits which are related to productivity, but also related to reducing the emissions in the atmosphere, and also related to water quality produced, then I think it could change quite dramatically, because what's driving a lot of these things is the economic prospects, not necessarily the environmental prospects.
I often wonder about that. Oh, sorry. No, okay.
Hi, I'm Anna Hislop from the Foundation for Arable Research, and I just want to turn one of those last statements around, because obviously as the arable sector, we're at the other end, so nitrous oxide is our key issue, less rather than methane, and we are working really hard, and our long-term vision is for mixed arable systems as well, bring more livestock in. But what we're constantly up against are these sort of perverse outcomes where freshwater policymakers don't talk to greenhouse gas policymakers. So a mixed arable system is going to provide you with a lot better soil quality, but it's going to increase your methane. And so we can, whichever way you go, it's really hard. So we're really happy to think about those sorts of things and work with anybody that can figure out how to do a mixed arable system that ticks everybody's boxes.
That's taking trade-offs to another level, but it is probably what we need to do. Is there any other comments, Graham?
This is David chuckling. Okay, plant-based protein, obviously it's a very peaky production worldwide. We say we're going to go away from our animals and things like that. Very easy for Australia, if they get a wee bit of rain, to produce a vast amount of grain. Same goes for USA, same goes for Russia. We could get buried in the world markets, would be an interesting question. Price goes up, price goes down. We are a very small country in terms of our area of arable, and yet we do have the world records for wheat production. So we can do some good things here for sure. Okay, methane, I think there's going to be a lot of money. Asparagopsis and other things that Lincoln University is working on, I believe that. Glyphosate resistance for no-till could have some big implications if we want to go to a lot of cultivation. We haven't talked about alternative mixed swards. I won't use another word that David doesn't like. But obviously if we cultivate paddocks, we're going to struggle with that carbon. climate solutions.
Thanks. One of the things that I meant to say at the end there was, I kind of talked about what Andrew had done in terms of pulling together lots of data from different sources and then modelling it at the national scale. One of the things that I think we set out to do in this program a bit was to go from the microbial scale through to the paddock scale and link that all up in a modelling framework. We kind of have gone from the lysimeters and the eticovariants, but it's actually linking those microbial processes and the work that's been done in that space by Gwen and Andrew and Andrea, and then linking that through to the ecosystem processes or the functions that we're interested in is something that we haven't fully cracked. And it's, I think, a gap. You could have debates, which I've had with Miko, around whether or not we need to do that. But yeah, that's something that I think a lot of people are striving for, and it could be an avenue for future research. I'd be interested if anyone had thoughts on that, because we've kind of, we've done quite well at scaling, but we're upwards, but not right down to that real, into those functions. Any other?
Yeah, maybe in response to the challenge that Paul put up there. I mean, any analysis and modelling in particular is really always related to the questions we're trying to address and trying to answer as that. And sort of something we've talked about in yesterday and today is the extent to which these very fine-scale processes do play an important role in terms of understanding the factors that we're ultimately more interested in. So, like say, greenhouse gas emissions under different management regimes. Can we predict that or can we better predict that if we know about the microbial contributions, differences in microbial components or not? And so like we talked about it yesterday and what I heard today, I kind of fail to see the evidence that the consideration of these microbial details does materially help us to better understand what happens at a larger scale. So the key questions that we're trying to address, we seem to be able to address them just as well or not as well, but the microbial components don't seem to really help with that. So I think that's an important part in terms of setting a future agenda, whether it does help. We have quite a powerful methods now to give us more information about what happens at the microbial level, but does it help us understand what happens at the levels where we really want to know the answers? And I questioned it before today, and I don't really see the evidence today that would have overturned that in an important way. I think there is still a need to try and scale up and better understand that, but it's also a very difficult task to find the right level of integration where people can still get an understanding of what's going on without creating lots of black boxes where nobody quite knows what goes in and can't really understand why the answers come out the way they do. But I think I'm not quite sure how you see it, how what's come out of this 5 years of work here in terms of those generic understandings.
I guess I'm just thinking of Andrea's one. It was a very clear separation in the microbial communities and some of the functions. So we could make a link that they were important. It's probably taking it to the next level is of what that actually functionally means. And in that case with plantain, it has been done to some degree. We still don't understand all the mechanisms. But yeah, it's maybe just knitting all that together a bit better could help.
Go Gwen.
Go Gwen.
what Rich said, and then that
just
sparked
Gwen.
conversation.
Gwen,
Can you hear?
just start again. I think we missed the start.
Can you hear now?
Yep.
Yeah, okay. So I'm just going to step back to what Rich said and then fast forward to what Miko's just discussed. So for me, the biggest headache I've had with this program and other things I'm trying to do is that we know that there is a big, it's almost like a busload of changes that are coming our way. And they are coming through regulations, the public perception, but also, you know, I know that some of you don't really like the concept of regenerative agriculture. You know, I'm really interested in that. But Nestle last week announced that it's expecting all of its commodity suppliers to adopt regenerative agriculture, and one of the biggest clients for Fonterra is Nestle. So there's going to be a lot of changes that are going to be pushed upon us in New Zealand. And where I'm getting the headache is how as scientists do we shift the way we do research to provide the answers that are required. So this program is amazing, but it costed millions and millions of dollars and took 5 years. If we are to do the same kind of research using the same kind of approach on each of the different innovations that could potentially meet the need of those customers of ourselves and how we are exporting our food produce, how are we going to deliver that research? Like, clearly what we're doing right now is not really sustainable in terms of the amount of money it costs. The second thing that leads on to the microbial part of it and the role of this small biodiversity, I think in this program we really, initially our aim was to try and decipher the processes so we could then plug them into or work together with Joe and build them into the model. And that was really hard to do because that would have required a lot of changes in the model, and that would have required also a lot of work on our part in terms of all the analysis we would have to do, and it would have become like a big massive mountain. So in that context, there was a limit to what we could do to answer questions that are driven by trying to decipher processes. But there are a lot of work that are ongoing elsewhere where microbes and the small diversity is used as bioindicators that are much more integrative of what the whole system looks like. It just takes a mind shift to think about them in a different way. So that's what I wanted to contribute. In terms of the land use change from dairy, there are some really interesting initiatives happening. There's, I don't know if you know of the company Lift Food that is trying to optimize growing crops by dairy farmers, and so just modify the way they grow, say, their winter crops, but for the sake of extracting plant protein, and they are currently looking at how they could do that using no-till and methods that conserve carbon, for example. So there's quite a lot of opportunity out there if we change the way we do science to get those numbers behind what these guys are doing. And there's loads of innovation out there that we can start studying. It would just require us to do it in a different way because we don't have the resources to deploy so many $1 million for each of the different initiatives that are coming up. And I'm interested to see how you all collectively think that could be done. Thank you. That's me.
Thanks, Gwen. Lots to take in there. But if I summarise, Rich is saying there's a lot of regulatory drivers coming that is going to potentially push land use change as a requirement to meet the limits. And then Gwen's emphasised there there's definitely a consumer drive or products marketing drivers coming as well, and they are both linked to some degree. And so we are going to have to change land use and land management. Has anyone got any thoughts on what Gwen said around accelerating some of the science that we do to get some answers more quickly and more cheaply? And I'm just having a thought there that some of what we've done here has been very, what would be kind of digging quite deep with some of the PhD students and looking at the mechanisms, and I think we need to do some of that research just to move the frontiers forward, and then some of it's been a bit more applied, so maybe there's a balance in there. But Be interested in anyone else's thoughts.
You ask Murray to make a comment as sorry, Murray, sorry. That's just going to be
Yes, Murray Doak with MPI based in Christchurch. And I was thinking, well, we are thinking the same thing. We look after the Islamic Fund, we look after the Sustainable Fibre Futures Fund. In particular with Islamic, we are thinking, what is our 5-year plan? And part of that is, what are the gaps in research, or what are the gaps in being able to fund particular types of research? And we're kind of opening that question up, really. Is it a problem in the structure of research funding that you guys can't get the funding you need to do the type of research that might be a bit more practical? I don't know, but it's where we're thinking, what's missing here? Because Richard drew attention to those things. Right at the start, there was, the aim of this project was to create, or to understand better how we could get a 10% improvement above best management practice. So, has this project done that? Do you think? Have we got some science that could do that? And therefore, what's the next kind of bundle of research? It might be multidisciplinary research. Can that get funded, or is it just that nobody's put up the projects yet? Because SFF is about that. Islamic is more about science research funded by the government with no need to come up with contributing funds. So it's, it fits more of a science model. So just some thoughts to throw at you.
Thank you.
It's a question from the audience that kind of builds on that. So maybe, oh no, they want me to ask it. And maybe David might want to comment as well. But Alison Slade has joined us from MB, and she's interested in how well she feels the program has met its original aims, and how you intend to get the information out more broadly to generate impact for New Zealand.
Thank you, Alison. Thank you for asking the question. We look forward to meeting you someday. I believe you've just taken on the role as manager of these programs. I would argue that we have clearly met the scientific objectives of this program in terms of we've delivered. We've delivered in terms of science and credible recognition, both nationally and internationally. I would argue we've also delivered in the sense of practical solutions. We've provided credibility for practical solutions, and we provided recommendations for farmers, and that of course is particularly around the winter activity growth and the possibility of using different crops to manage. So effectively, I would say that we, in fact, if all of those practices were implemented across dairy in Canterbury, which would be difficult to do, but if they were, then I would say we would decrease nitrogen leaching by more than 10%, which is what the original aim of the program was. Now, we would now argue that that has had an enormous environmental benefit. We would no longer go back to the question that the issue that Paul raised, that it would give us headroom to actually establish more dairying without any extra nitrogen. So I believe we have achieved that. Now, when it comes to the adoption of these practices and the encouragement of people to put these into practice, I think we do really have to argue that these are longer-term outcomes of the program, which will only actually occur beyond the life of the program. And it is still up to us to publicise and to promote. And what we've been doing is that in the 5 years, we've had an unmeasurable number of field days at Ashkenazi site, mainly driven by Lincoln University, but we've always been there to show what we've done as well. and we've told many people about our findings and our recommendations. So that word is getting out there. But we're not going to see the implementation of all of our recommendations by next week when the program ends. That's going to have to come in terms of the long-term outcomes.
Thanks, David. I thought that was a nice summary. Did you have something?
I never know else. Can I just say one thing?
Sorry, I've got to run, but while we've got MPI and MBE on the line, PhD students and postgrads are the engine room of research at universities, and at the moment we're not getting any into the country. And there's going to be a big shortage of research that occurs as a result of that. So the sooner you can get them in, the better.
And you could see from the presentations, they did some pretty cool research in this program. Some of the really, I don't know, like I said, fundamental cutting edge stuff. It's like, well, that's new, that's interesting.
Just before we dance back to Gwen, Alison's got another question. Sorry. So Alison is only 3 days into her role, so she can comment on the funding question, if you've ever heard. But she has asked you, David, is there a role for SLMACCN?
I think extension, kind of the extension for SLAMAC.
Well, yeah. Well, we have Murray here who can perhaps comment on the SLMACC. But yeah, I mean, effectively, the one good thing is we spent an enormous amount of money and time setting up those facilities. And when a program comes to an end and funding comes to an end, it's incredibly sad to say I'm going to pull the plug and walk away and leave and not do anything with it. The opportunity we've had is to take those lysimeter facilities, with a program led by Scott and colleagues in Hamilton as part of an SLMACC program. And so we are continuing to use that, and we're continuing to use the data for the last 4 years to inform the SLMACC and MPI of our findings. So effectively, it's ongoing. It's continuing to living on, and of course, it's always felt that the SLMACC side of it would really help to look at more the application of the work. But we would never have been able to persuade SLMACC to fund the work that we did on those fundamental processes. And in some ways, that's where the issue comes. I mean, we literally have the choice of Marsden or MB Endeavour, really, in terms of, well, we obviously have our own SSIF money, but that never stretches that far. So it's actually getting the right mix of new innovative science versus applied science that's going to produce an answer at the end of it, and tailoring the proposals that we write to match those particular funds.
Alison, if you just want to unmute yourself.
Yeah. Can you hear me?
Yeah.
Hello everyone. For those of you who don't know me, I have up until about 10 days ago been employed by Science, so I'm very much aware of the problems around a research program being translated to impact, and it's an area that I've been very interested in for some time, and I've come into the Endeavour Fund. I am currently the second on the Endeavour Fund. It's a question that I want to raise with an MB because of this issue where you've got this fundamental research program which does have quite a strong element of applied science, but once the funding stops, and what happens then? So it's not something I've got an answer for, but it's a conversation that I would like to drive with an MB because of the experience that I've had. Firstly, 20 years of the technical end in research, and then 12 years in research management. So I'm just listening to the conversation with interest, and I will take the conversation back with me to MB. And as I say, it's something that I'm going to be really thinking about and talking about while I'm there. So yeah, just thought I'd let you know that. I'm very interested also in that connection between this sort of research and the SLMAC fund, particularly because being a tech transfer statutory type fund, it seems like an obvious way for some of the work to go. So yeah, just letting you know that I'm on to it, and I hope I can make some progress while I'm there.
Thank you. That would be really good if you could do that. And I actually just reflecting as you were talking there and others have talked, I think this is actually probably quite a good program where it has fed into a range of other programs. Like we think it's linked through to Carbon Watch, it's linked through to the NZAGRC, and then it's gone into we've got the SLAMAC work, which by definition in the proposal they wanted something that was proven already and just getting validated good numbers around it. So it was very applied and getting hard numbers around something we knew rather than cutting edge science. So it's got a lot of touch points to a lot of other programs. And I think the key is, especially for some of these where there's infrastructure with a program, having a pathway to utilise that when the program finishes, I think is pretty valuable. Like we were lucky in that we were proactive when Scott put a proposal in and got the lysimeters funded, but it would be a shame if that had just completely stopped and we just couldn't do anything, because they're probably $1 million or more to get set up, and we've got some really good data because of their size and on those unique soils.
Just a quick response to you, Alison, if you can hear me. I've put a little bit of effort into this impact and I guess question over the last few years, and I guess experience has kind of taught me that the greatest impact is derived by those programs who put about 10 or 15% into monitoring and evaluation. And it's something we do really, really poorly in our we should be doing that from the offset to know how much of a difference we actually make. And it also enables us to be agile and change direction, filling in capability gaps when required, but also, importantly, taking a regular map of our stakeholders, those who are of interest, but also, importantly, have influence and are able to make change. So if you can get hold of those people, you not only get their ear, but you can also, to some degree, handle the issue of legacy and stewardship that David was talking about. And I know it's a damn shame that science just gets left, but if you get it with the right stakeholder, it can have a legacy.
Cool, thanks Rich. Miko's got another question, comment. And if anyone else that hasn't commented wants to, your opportunity is after Miko.
Thanks. Yeah, it's more comment. It's sort of, this program is, you call it reducing nitrogen losses from plants, and all the emphasis has really been in on what happens when nitrogen reaches the soil, and useful insights have been gained with that. And we've seen some of the results, like what William talked about earlier. It really, a lot of the fluxes are really related to the excess nitrogen that comes to the soil, and I think we need, of the really promising approaches is to try and minimize the nitrogen that comes, particularly in dairy systems, the nitrogen comes to the urine spots in the first place. That's where the problem starts, and that is something that where we have much greater scope to reducing that in principle by reducing the nitrogen content of feed, which then leads to less nitrogen excess in the cows, leading to less urine flow to the environment. And there is really a lot of scope, and I think everything I've seen, whether people actually directly looked at it or not, but the results that people find is that reduced nitrogen content of feed leads to less nitrate leaching, less nitrous oxide, whichever way you try to look at it. And I think where the real opportunity lies for the future is to really target that. See how far can we push the system to try and operationalize the management of the nitrogen content of feed to really reduce that excess, which then starts the whole cascade of problems. Just looking at it at the end, once we're putting the ambulance at the bottom of the cliff, and instead of seeing whether the problem started further up, and there seems to be a lot of potential, a lot of scope to minimize the problem by, in fact, a lot more, reduce the problem by a lot more than 5 or 10%. If we can just manipulate that nitrogen content a little bit. We're doing that to some extent without explicitly focusing on that to plantain. That gives us a lower nitrogen content, as well as possibly other things. But that's probably the most important part to which plantain has the beneficial effects. But there are other ways by which we can manipulate the nitrogen content. And a project specifically focused on that would be my best bet at this point to work some really gains into the future.
Thanks, Miko. And I was just thinking about that. That's very focused on a pastoral kind of system, but maybe the comment before around more integrated systems is an opportunity to bring in some of that lower nitrogen feed from cropping systems potentially. I'll go to David, and then any other final comments from someone that hasn't said anything, and then back to David to wrap up, I think.
Okay, thank you. I just wanted to raise the issue of technology. I mean, we are getting increasingly capable now in terms of technological advances. And we haven't talked about this today. What I'm talking about is, it's not one size fits all, is it? We have to match climate, soils, and land use. And we need to be able to find techniques, or we have the techniques available to us, to be able to define what the limits are on those different places. Some of it's more advanced than others. We do have variable rate irrigation. That's not practiced at the farm at Ashley Dean at the moment. And maybe it doesn't need to be where we are because it's all the same soil type. But when you're looking at growing different crops, which require different amounts of irrigation at different times of year, we don't want to just irrigate them all constantly the same. And it's this, and the issues of carbon, in terms of some soils are going to obviously retain or lose more carbon than others. So they're more sensitive, they're less resilient. So we have to tailor our management practices to suit the local conditions, but be able to extrapolate it at the landscape scale.
Thank you. Is there anyone that hasn't made a comment that wants to add any final reflections? No? Everyone's tired out?
I guess it's just going back to the results we got from this. I think there were some really good results with nitrogen losses, and you see like the plantains going on to DRNZ, DCC funding for that plantain proficiency. And I also think one other big thing that came from this project is the training up of people, like I think myself, Andrea, Jonathan. The skills that we acquired through this program I think is really helpful when we go out into our careers, into the industry. So I think that's another big output of this project, is training up people like myself, young scientists as well. I think that's a really big output of this program as well. So thank you, David.
Thanks. Any other comments? Oh, looks like I've got one online.
Gwen's just asked me to convey her comment. Along with M&E, what is also missing is funding specifically set aside for designing collaborative projects. The NEC are funding this type of initiatives, but MB and MPI are not doing so enough, which means large programs might get funded, but the legacy, connection with stakeholders, etc., are set up in a shallow manner due to lack of funding.
Yeah, I think that's an interesting comment, and I could reflect that too, that often there's only so much you can do with co-development with stakeholders if you're doing the proposal all yourself before you've got any funding to do anything. You can only do so much. Shall I hand it back? Any final comments from somebody else that hasn't said anything that wants to add something? Otherwise I'll hand it back to David.
Thank you, Paul. Well, it just remains for me for an enormous thanks to you all, really. First of all, thank you to the people who have stayed with us all day online or came in and out. We didn't get to meet most of you, but there's plenty of opportunity to keep on talking about these aspects. So thank you for Thank you for being part of it. Thank you to everyone here in the room, those who gave presentations and those who added to the discussion. But of course, thank you to the 30 or 40 people involved in the research over the last 5 years. It's been an amazing effort, and I think we today have seen the benefits. We've seen the opportunities, and we've seen the value of doing this sort of research in terms of where it's getting us. There will always be need for more research.